I'm really pleased to introduce Dr. Malyon Bimler today as our guest speaker. She's a postdoctoral researcher at the University of Melbourne, working with Dr. Margie Mayfield's lab group. And her work explores how fundamental features of ecosystems like resilience, stability, and biodiversity emerge from the complex web of interactions that occur between species. Her work is particularly interesting as she focuses on biological complexities like plant-plant facilitation or nonlinear interactions that are often ignored because there's not clear theoretical expectations. So her work acknowledges this complexity and is building toward a more inclusive theory of biodiversity maintenance. So I'm personally very excited to hear from her as one of her papers on the role of facilitation in modern coexistence theory has really shaped how I'm approaching my dissertation questions. Yeah, so I'm really pleased that you're here, Malyon, and I'll turn it over to you here. Thanks for giving up your Saturday morning to be with us and share your work on what plant interaction networks tell us about coexistence. Thank you.
Thank you so much, Carmen, and thank you all for coming today. I'm sorry about the little technical difficulties we had. Luckily, I tend to speak a little bit faster when I present than when I practice, so hopefully I can fit everything in. So you'll notice my talk title is a question. Just a word of warning for anyone who's hoping for a nicely, neatly packaged answer at the end of this. I unfortunately don't have one. What I'm going to be presenting today is a collection of studies that really raise more questions than they answer. And hopefully you leave this as excited as I am about trying to figure out what this all means. So next slide, please. Thank you. Just for a little bit of context, so I'm a postdoc at the University of Melbourne. That's the most southern little star down there in the Mayfield lab. I actually live in subtropical Queensland, which is up on the right. That's far more subtropical, very, very humid and wet kind of climate. And then the system that I'm going to be talking about today, the system I've used to test a lot of these ideas, is in southwest Western Australia. So that's a very dry and arid Mediterranean climate. It's heavily agricultural. It's the Australian wheat belt. And so in between these vast fields of wheat and canola, you have some remnants of woodlands or of Australian bush, as you can see in the photos. Those are usually a very sparse canopy of predominantly eucalypt and acacia trees. And during most of the year, about 9 months of the year, it's complete bare dirt underneath. Next slide, please. Oh, next slide, please. Oh, one before, sorry. The ones with the photos of the system. So anyway, it's bare dirt most of the year. There's winter rains around July and August. And so this is the start of the growing season for annual wildflowers in the system that pop up under this mixed sparse canopy. And these are wildflowers that grow and set seed in about 3 months from September to October. When they are growing, they're very, very diverse, as you can see in some of these photos, and they also have very patchy distributions. And so we already know in the system that we have very variable interactions between these annual wildflowers, that those interactions vary according to the environment. And so in turn, we have variable coexistence outcomes. So what I mean by coexistence is the ability of species to persist together through time, despite affecting one another or despite interacting with one another. So we have variable coexistence outcomes in this system, and that some of these wildflowers are predicted to coexist in some areas of this environment, the study system, but not in other areas. So it's very complex and diverse, and to understand and predict these patterns of coexistence and composition and distribution, we need to look at the interactions that are occurring between these wildflowers. Next slide, please. And so interaction networks have been really quite well explored for a number of systems. For example, for food webs, for plant pollinator systems, or for host parasite. A network is simply a description of who interacts with whom, and in some cases, whether this interaction is beneficial or harmful, and how strong the interaction may be. Next slide. And so we can summarize networks through various metrics that tell us about how the interactions are structured in a community. Here you can see a network on one side and the matrix on the other. Just in case it's unclear, a matrix and a network are essentially the same thing, just different ways of showing the same information. And so the kind of things we look at when we're looking at the structure of an interaction network is, for example, whether the community is tightly connected or whether it's more loosely connected. Do species cluster into modules that preferentially interact with one another or not? Really, to summarize, the network approach gives us quite a holistic view of the whole community and all the species that are interacting within it. And so some of these different metrics and ways of being organized promote stability, it promotes resilience, or it promotes diversity. So essentially, interactions in a network or a community are structured in ways that affect the maintenance of diversity and boost coexistence. And empirical food webs often in ways that actually promote this diversity and stability and resilience. So I've given you a quick overview of networks. Next slide, please. But what do we mean by plant interactions? So when it comes to plants, interaction networks have received far less attention. That's due to several factors, but one in particular is that it's very difficult and time-consuming to measure plant-plant interactions. So unlike food webs or plant pollinator networks, where you can go and directly observe who is interacting with whom, we can't really do that when it comes to plants. Next slide. So what do we do? How do we measure an interaction when it comes to plants? So typically, we measure interactions as effects on performance of a focal species across varying densities of an interaction partner. So I say performance, that can be anything related to fitness, that can be biomass or growth or survival or fecundity, etc. And so in this example, the little pink plant species, Scarina, I'm just gonna call it the pink plant, is growing by itself in one, and then you see that it's growing in low density of the yellow plant and high density of the yellow plant. And so the interaction that we measure is the average effect on performance compared to when you are growing alone. And so we use the little experiment in 1 and the little experiment in 2 to fill the entries in the interaction matrix in 1 and in 2. So you don't need to worry too much about the actual number I've put in there. It's just indicative. It's just to show that in this case, pink plant has a mild competitive effect on the yellow plant, but the yellow plant has a much stronger competitive effect on the pink plant. And once again, these effects are effects of a neighbor on the performance of a focal individual. Next slide. So from these kind of experiments and from plant population ecology, we've learned that plant interactions are predominantly the result of resource competition, so competition for sun, for water, for nutrients, for shade, so forth and so on. And so essentially we found that plants tend to perform better when they're growing alone under lab conditions, and sometimes even under field conditions, than when they are growing with neighbors. So in a lot of situations, having neighbors is not so good for you, and you are bigger, stronger, more fertile when you are growing alone. So this is under resource competition. And obviously, so all plants compete with one another in this situation. If that's the case, then some plants are going to be better competitors than others. And so under competitive conditions, plant communities tend to be structured into competitive hierarchies, where you have one plant that's the best competitor out of all of them, it outcompetes all others, and a second plant which is almost as good, but not quite. So when we think about coexistence, what does that mean for coexistence, and how are the competitive dominance kept in check? Next slide. And so one crucial aspect of coexistence in competitive conditions is the idea of negative density dependence, or in other words, competition with yourself. And so in this little interaction matrix, those are the values on the diagonal. So you can see that pink competes with itself more strongly than it competes with yellow, and same with yellow, competes with itself far more strongly than it competes with anyone else. And so this basically means that any competitive dominant is kept in check and doesn't ever become overly abundant. And so this is one of the basic principles of coexistence and how it can arise under competitive conditions. And next slide. Or perhaps rather how coexistence can arise despite competitive conditions. So this maybe how we conceptualize plant interactions, but in reality, plants actually interact with one another through many, many mechanisms, not just competition for resources. Next slide. So I won't list all the examples here, but the effect of one plant on another's performance is the outcome of many factors. Obviously, resource competition and the abiotic environment, but also other things such as attracting pollinators or repelling herbivores. And some of these mechanisms are going to be beneficial to a focal plant. Some of these are going to be harmful. On top of that, many of these mechanisms are variable with time and space, and they're dependent on the abiotic and the biotic context there. They're dependent on the environment and also the presence and absence of other plants and members of the community. So for example, here perhaps pink attracts lots of pollinators, so it has a beneficial effect on yellow, but if there's many, many other species or other wildflowers around, then that dilutes the effect of pollinator attraction. So even a mechanism that might occur predominantly between two species can also be affected by the whole community. Now, perhaps you see where I'm going with the use of networks. Next slide. So as a result, the overall sum of all these mechanisms that mediate the effect of one plant on another plant's performance are incredibly context dependent. And so what might be good under some interaction that might be beneficial under certain conditions might be harmful under other conditions. Next slide. And so perhaps our little focal plant, our little pink plant, produces a lot of seeds. Those are the little badminton things on the side. So perhaps they produce lots of seeds when growing next to this poor neighbor in a dry and sunny year, but they'll produce far fewer seeds when growing next to the same neighbor under wet and rainy conditions. And here I'm talking about seeds because seed production is is our measure of performance that we use in our wildflower system. And next slide, please. So of course, we are well aware that interactions are context dependent. And so we deal with this by measuring an interaction as an average effect. That's why we average things. So we can take these two different conditions, sunny year and wet year, and we can average them, and we compare them to when a plant is growing alone. But if you're good at counting C's while I was talking, you'll notice on the slide that taking the average can actually mask the true effects, or perhaps more subtle effects and dynamics that are occurring between our little plants. Next slide, please. Okay, so now this is a result from a paper that came out, well, quite a few years ago now, but this was a paper that kickstarted a lot of this interest in networks and in facilitation. And so in this particular paper, we were measuring interactions as effects on seed production between our wildflowers of Western Australia. We grew focal species under varying densities of neighbors, and we also measured environmental conditions, in this case, tree canopy cover, which tends to structure the composition of the community quite a lot. And so instead of taking interactions as just an average across the whole environmental conditions, we model interactions as a function of the environment. And that's what you're seeing on the figure right there. On the y-axis, you have the strength of that interaction. Anything that's above 0 is competitive, and anything that's in the gray area is facilitated. And so we found that interactions between pairs of species were indeed a variable. And as a side effect of wanting to model an interaction as a function, we allowed this interaction to be facilitative, to take on facilitative values, which is not something that's typically done. And this was not what we'd initially set out to investigate, but we found that there was quite a lot of facilitation. And that even if the interaction on average was competitive, there were areas of the environment where the interaction was quite clearly facilitated. Next slide, please. So this means that if we try to predict whether species will coexist or not based on an average interaction value only, we're very likely to make inaccurate predictions. And the results from this paper also challenge the assumption that plant communities exist only under competition. And in the case of our study system, it appears that species are persisting together through time under conditions that include both competition and facilitation. But it's very difficult for us to make any predictions or to know even more about what that might look like, because the vast majority of our theory is built around resource competition. And so we don't know a whole lot about how facilitation interacts with competition. And so for this paper, we found also that a variation in interaction across the environment was very idiosyncratic. This is just a figure for one specific interactions, but there were all sorts of different shapes that were present. And we realized we needed to find out more so that we could make some generalizations. For example, was facilitation really that prevalent, or was it just a fluke and we chose 4 species that were very facilitative? Is it just limited to certain extremes of the environment, so forth and so on? Next slide, please. And so we decided to look still at plant-plant interactions in the system, but to increase the number of species considered. And so in order to do that, we had to reduce the grain of environmental variation. So we could no longer measure an interaction as a function of the environment. Instead, what we did is that we grouped our environment into 3 categories. That's what you have on the right: sunny plots, intermediate shade plots, and heavily shaded plots. And so we split the data up into these three categories. We averaged interactions within each category to have 3 interaction networks, 1 for each environment. And just as a note, this is far from perfect because we're still averaging across an environment, but we're, well, at least accounting for some variability. And we once again allowed interactions to be competitive or facilitative as dictated by the data. Next slide, please. So this kind of project can be very data intensive in order to have enough observations to be able to reliably quantify interactions between many species. We had, well, very small plots, 50 by 50 centimeters, and 100 of them. We limited the interaction neighborhood, i.e. what you count as a neighbor, to a radius of a few centimeters from a focal species, and that's because these are really, really tiny, incredibly small plants. We still measured interactions as effects on seed performance, and we used naturally occurring variation in density in order to estimate those interactions. We also did do some thinning treatments to reduce density, but not diversity. So we thinned a quarter of plots to 30% and a quarter to 60% and left the rest unthinned. And that helps remove any confounding effects of environment and neighborhood density. So we measured seed productions for focal individuals belonging to the most common species in this system, and the identities and abundance of neighbors within that small interaction neighborhood to get interaction effects on performance and fill this interaction matrix on the right. Now, because we're dealing with naturally occurring densities, some combinations of species aren't observed to co-occur or not at variable enough densities, sometimes simply by chance because we've got some rare species in the system. And so we don't have the observations to be able to estimate those interactions. Next slide, please. So this is quite a common problem, which has many different solutions. The one that we picked is that for those missing interactions, like let's say the one between the effect of yellow whites here on pink scarinus, we make a different set of assumptions that allows us to still make use of this data we collected and produce some kind of estimate. So we may not know the effect of yellow on pink, but we know from the data set we've collected how yellow tends to affect other focal species. And we can use that to calculate an average effect of yellow on its neighbors. And likewise, we may not know how pink responds to yellow in this particular situation, but we know how it responds on average to its neighbors, and so we can calculate an average response. And then to estimate the missing interaction, you just multiply the effect of yellow by the response of pink. Next slide, please. So this allows us to plug the holes in the interaction matrix or the network and to estimate all interactions between our focal species, even when using data that was not necessarily designed to capture or observe all potential interactions, data that relies on natural variation in density, like ours, or in diverse systems where there's a lot of interactions to observe and quantify, and there's a lot of interactions that are potentially not observed. So all these non-missing interactions and the missing interactions are merged together into the one network or matrix. There's a method paper for this that's been published, and there's also code available on GitHub to run this on your kind of data. And so in our case, we had about 80% of interactions that we could observe and about 20% that we could not, though you can go much lower than that in terms of higher proportion of missing interactions. Next slide, please. So applying this method to our data resulted in 3 interaction networks from each of our shade categories, one in the open environment, a bit of shade, and a lot of shade. And so between those environmental categories, we did have variation in species composition, but plot level diversity was very similar. We had a total of 21 unique focal species, some of which appeared in multiple environments or multiple shade categories, some of which did not. We were mainly limited due to data requirements for species-specific estimates of seed and germination, because we like to transform our measures on performance or seed production into effects on future population abundances. And so for that, we need germination and survival rates. And so that's what limited us to 21 focal species. And so we had 3 different size networks, 12 species in the open environment, 14 species in the intermediate, and 17 species in the shady environment, all belonging to 21 unique species. The system itself is far more diverse than that. There's been over 80 species counted, like wildflower species counted at this particular reserve. And we were able to extract the interaction networks, which is what you see at the bottom there. That's just an example for the open environment. So all the little nodes are different wildflower species. The gray nodes are the 9 species that were found in all 3 shade categories. The ones that are circled were present in this particular one. And then there's some little plant names that don't have a circle around them. That's just because they weren't present in this particular environment, but they were present in the others. And so the arrows connecting all these little nodes to one another are our measures of interaction effects. Anything that is in orange indicates competition. Anything that is blue shows facilitation. And just as an aside, a lot of the interactions aren't actually visible on this particular graph because the effects are very, very weak. So this network image in itself doesn't really tell us much, but it does show that there's both facilitation and competition involved. Next slide, please. And so indeed, we found that most species emitted both facilitation and competition. So here we're looking at the output, so the sum of all the arrows leaving a species or leaving a node. If you sum up all the orange ones, you get the competitive output on the x-axis. And if you sum up all the blue arrows, you get the facilitative output on the y-axis. And the little diamonds are just the median for different species present in this intermediate shade environment. And so you see a lot of species emit competition and emit facilitation. Next slide. And this is true across all 3 environmental categories. Our next slide, please. Thank you. Some species, you can see, for example, in the middle graph, there's a pink diamond. That pink diamond is also visible in the shady figure on the right. And so you can see, so this is just a species that emits competition and facilitation, but the relative amount of each varies depending on what environment it's in. Next slide. I'm just going to rush through some of this. So likewise, a species also receive both facilitation and competition. So now we're looking at the sum of all the arrows that are arriving into a node, competitive for orange and facilitative for blue. Next slide. And once again, across all 3 environments, species tend to both receive competition and receive facilitation. Next slide, please. Now, if you remember the beginning of the seminar, I mentioned that we expect species to compete with themselves very strongly when they're under a competitive system. And this is how coexistence is maintained under competition. But does this expectation still hold when the system is not entirely competitive? Next slide. And the answer is that no, it doesn't. So I'm sorry, this is not the ideal figure to present, but what you're looking at is interaction with oneself of this graph, the grass on the right, Pentamerus aeroides. So this is how strongly it interacts with itself under open conditions on the bottom, mildly shady and heavily shady. And so the little red dots are the median of an interaction strength. Anything that is on the right is competitive. Anything that's on the left of 0 is facilitative. Sorry if the colors are confusing with that. I realize I'm repeating the orange pattern here, but essentially what you're seeing is that some species facilitate themselves instead of competing with themselves, and that this varies depending on the environment that they're in. Next slide. So far we've looked at how species behave in each environmental category, but what about across all these environments? And so, well, what we found is that both between species and within species, so for those self-interactions and for interactions with others, they're predominantly competitive, but facilitation is really not uncommon at all, and definitely far more common than what we had actually expected. How am I doing for time? Good?
Yeah, we've got some time left.
Okay, great. Next slide, please. So we've looked at the distribution of these interaction strengths, but we haven't actually looked at how the interactions are organized within the networks yet. How are they structured? And so I'm going to just explain to you a few of the metrics that we can use to describe the organization of interactions when there's both facilitation and competition that's going on. So under number 1, what you have is the level of competitive hierarchy versus the level of intransitivity. So competitive hierarchy, once again, one species is a competitive dominant and outcompetes all others, and the second species outcompetes most other species except for that first one. And then, so that occurs on a spectrum, and this level of hierarchy is at one extreme. And then at the other end of the spectrum, you have intransitivity, which is more like a game of rock, paper, scissors. So there's no clear winner of competition, but more A outcompetes B, B outcompetes C, C outcompetes A. So those are just two opposite ends of the spectrum. And in, well, competitively dominant systems, we do expect more hierarchy. The second thing that I'm going to be looking at are interaction loops. So that is how, well, two species interact with one another. They can be cooperative, where they both facilitate one another. It can be competitive loop, where they both compete with one another, or an asymmetric loop where maybe pink competes with yellow, but yellow facilitates pink. And then lastly, I'm going to be looking at modularity, and that's the degree to which species will cluster together, form clusters of more densely connected interactions. Next slide, please. So here's our scale of intransitivity at 0 to competitive hierarchy at 1. And I've marked out 3 little networks in red. And what you see is that they are predominantly intransitive. They're definitely on the well, more intransitive side of things, and not particularly hierarchical. Next slide, please. As for interaction loops, what we found across all 3 networks is that the loops were predominantly asymmetric. So pink perhaps facilitates yellow, but yellow competes with pink. That was a predominant interaction loop, followed by competitive loops, and then lastly, a smaller proportion of cooperative loops. I'm telling you about the proportion of loops because it's a really active area of research in ecological network theory. Right now, there's various papers that explore how the different proportions of loops affect stability, but we're still not entirely, the verdict is still out on how exactly that works. Some studies suggest that asymmetric loops, the top one, confer the most stability. But there's some evidence that suggests that under certain conditions, cooperative loops can actually help stability or species persistence. And even more evidence that perhaps it's actually a mix of multiple types of loops, competitive, cooperative, and asymmetric, that contributes to maintaining coexistence. And then lastly, so I looked at modularity. So if there's anyone here who looks at modularity in food webs or in other systems, it's slightly different when you're looking at plant interaction networks because there's competition and facilitation going on. So specifically the measure of modularity that I used quantified to what degree species that are facilitative tend to cluster into little modules of facilitation. So species will facilitate one another in a module and compete more strongly with anyone who doesn't belong to their very own module. And what we found was that modularity was really quite low. So we did not observe this kind of clustering of facilitator species together. So just as a recap, the networks were predominantly intransitive, low modularity, and a lot of asymmetrics interaction loops. I scratch your back, but you don't scratch mine. So to conclude, or coming close to the conclusion, even if interactions themselves varied and the species varied from one network to another, the networks were overwhelmingly similar. Predominantly competitive, but some facilitation and the other metrics that I just told you about. Unfortunately, so far we only have these three networks, though there are currently other efforts going on to apply this method to tree plots. Those results aren't yet published, but they do suggest that facilitation is not uncommon in other systems as well. So it's not just a fluke of our little wildflowers. But essentially, well, this group of studies leaves us with a lot of unanswered questions and currently few generalities that we can draw yet. But clearly, coexistence doesn't only arise from purely competitive dynamics. And perhaps some of the features that are described here may contribute somewhat to coexistence. Next slide, please. So we may have a good understanding of how coexistence can arise under competitive conditions, but not when there's both competition and facilitation. Next slide, please. So yes, lots of unanswered questions. Do these results extend to other plant systems? How do we account for variable interactions? What do these network metrics all mean? Quite a few that I will let you ponder and ask if you would like. Next slide, please. So I would like to just thank my collaborators and lab members from the Mayfield and the Stauffer lab that I work very closely with. Thank you all for coming, and I am glad to take any questions. Thank you.
Okay, great. So happy to take any questions from the audience live. And if you're on Zoom and you have a question, you can feel free to raise your hand. We'll just call on you and you should be able to unmute and just ask a question that way. Anybody just Jeremy.
Hey, Malyon, that was a great talk. Super exciting to see all the work. I worked a little bit on these context-dependent interactions, and I've thought a lot about Like what sort of functional form should these functions take to try to describe these context-dependent interactions? And you showed a few examples. One that was, I think, like monotonic at the beginning, or sorry, monomodal. And one that was kind of like a linear towards the end of like interactions just became more competitive the more shaded you were. And I've found myself frustrated with kind of like the idiosyncrasy of it all and found myself somewhat pessimistic about if we're going to find any general theory of what should interactions look like along environmental gradients, how should they change? Maybe if we like understood all of the traits in the world, we'd know. But I was curious about your thoughts about, are you optimistic about like a pragmatic theory about interactions along an environmental gradient? Like, are we going to get to one that's satisfying and nice for how should interactions change along some environmental gradient?
Thank you for that question. It's something that frustrates me a lot, and I guess it kind of points to this constant tension we have between generality in models and complexity and being able to account for the situation at hand. I think that currently we're at a stage where we don't know enough about to be able to draw generalizations. As you said, the interactions can change across the environment in many different ways, and not just across the environment, but change with density and with the presence of other species as well. So it can be a bit difficult to come at this and understand how we are supposed to account for this measure of complexity. For now, I would say that I think accounting for any kind of complexity is good, and helps us perhaps eventually get to some more broad generalizations. I mean, I don't think we're gonna necessarily be able to say an interaction is overall going to be competitive under shade, but facilitative under the sun. I think that perhaps too variable and context dependent than that. But that's where I found actually the results here quite hopeful, because when you look at it from a holistic perspective, so the interactions themselves might change, but it may be there's always a certain proportion that's competitive or facilitative, or it stays within a certain range, or there's other aspects of the network structure that are consistent regardless. And so that's an avenue I'd like to explore some more, to try and yeah, find those kind of generalizations.
Great, that's very exciting. Thank you.
Thank you.
Other questions here or on Zoom? Oh, Lauren, go ahead. You can unmute yourself, I think.
Hey, this was a great talk. Thank you so much. Really appreciate it. Saturday morning push. And kind of we've been thinking a lot about interactions and also spatial and temporal scale. And I just wonder, at the scale that you're looking at this, and you're still seeing this really big balance of facilitation, but if you think that would change over say longer time or spatial scales, and I guess what I'm getting at is like what do you think the mechanism is for the primary competitive versus facilitative interactions? And do we think that by being at this kind of local scale, we're underestimating one or the other?
Yeah, so I would expect things to change, depending on if you're looking at smaller spatial scale or larger spatial scale, and same with temporal scale. I honestly don't know enough yet to be able to say how I think that might change. We have a PhD student in the lab, Courtney Taylor, who has just started a series of experiments to try and untangle the effects of spatial scale, and perhaps a master's student jumping on soon. So hopefully we can have some answers in a few years. I do think the mechanisms are going to change, No idea if the balance or the proportion of one or the other will change too.
Cool. Thanks.
Anyone else? No. One from the audience here.
I appreciate that you guys went out and did these kind of observational studies where you weren't actually seeding. I think that's correct, right? And so I'm curious if you feel like phenology is impacting this a lot, like if you're missing germination timing or if you see a lot of variation in flowering time, like is there any differences between shade and sun and those things that would be moving things towards facilitation or competition?
So yes, there's absolutely are differences in phenology, and I do think they're important. In this particular system, though, everything flowers and grows at very similar times. The whole growing season is really 2 months, maybe 2.5 months. So everything tends to, or even maybe 6 weeks sometimes if it's a particularly short growing season. So I would think that at least in this system, it perhaps doesn't play as much of a role as in other systems. But yes, I do think phenology is somewhat important, and we are missing from this any idea of how variation in germination and in seed survival affects things, and it's very likely that both of these things induce even more variation as well, more variation in interactions and interaction outcomes. So another layer of unexplored complexity.
All right. Well, if there aren't any other questions, let's thank Malyon one more time.
Thank you, everyone.
And I think she's slotted. She's got some time to speak with folks next week once she gets a chance